And welcome to our webinar today on MALDI-MS-based assays for cellular drug action and transport with rapifleX multi-pharma pools. My name is Anne Prof and I will be your host. If you should have any technical difficulties during the presentation, please use the chat box and let me know. At the end of the presentation, we will have time for a question and answer session. To ask your questions, please use the Q&A box. If you run out of time and are not able to get to your question, we will contact you afterwards offline. We have two speakers today. Our first speaker is Professor Dr. Carsten Hopf, who is the head of CeMOS. Professor Hopf heads the Center of Mass Spectrometry and Optical Spectroscopy in Mannheim, Germany. He is a professor of bioanalytics and drug discovery at Mannheim Technical University. Carsten obtained his PhD in biochemistry from University of Tübingen in collaboration with the Max Planck Institute for Developmental Biology. After Carsten's presentation, we will also hear from Melissa Unger, who is a scientist at CeMOS. Melissa Unger received a B.Sc and M.Sc in biology from Würzburg University and specialized in human genetics and pharmaceutical biology. She just submitted her PhD thesis about MALDI-MS assays of drug uptake and drug-drug interactions to Heidelberg University. And now I'd like to pass over the presentation to our first presenter, Professor Hopf.
Welcome, everybody. Before I start talking about cell-based assays that we run with the rapifleX MPP, I would like to briefly introduce our CeMOS Center for Mass Spectrometry and Optical Spectroscopy in Mannheim. We are a center of about 60 people who do co-development of devices and IT with various types of companies, so a very industry-focused approach. And we also co-develop applications and ITs for various sectors of the industry, ranging from chemicals to life science. And we have the pleasure of operating a center fully equipped with Bruker instruments. And as you can see, in the middle of the lower picture, there's the rapifleX that we will focus on during this talk. And briefly, before I go into cell-based assays, I would like to introduce to you the enormous speed of the machine and also the general operating principle of MALDI-MS spectrometry. The great feature of a rapifleX mass spectrometer is that as it scans over target plates or even tissues, it acquires mass spectra on the fly at an incredible speed. And at every position, the laser ionizes a complex mixture of biomolecules that are then separated based on their mass-to-charge ratio and that hit a detector at different times of flight. And based on those times of flight, we can reassign the type of biomolecule that we are looking at in this very high-speed application. Together with Melissa, who will take over in part 2, is to first talk about label-free drug profiling in phenotypic and mechanistic cell assays. And Melissa will tell you more about drug profiling of uptake scenarios into cells and how that is relevant for drug-drug interactions. So to start with part number, the label-free drug profiling in phenotypic and mechanistic cell assays. And indeed, it is one of the big advantages of the MALDI-MS spectrometry technology that it is label-free. And as those come from the assay development field, in particular cell assay development field, will certainly know currently the majority of assays is using label-containing methods, either being fluorescence-based or some still being radioactivity-based. And in particular, fluorescence readouts can have a couple of distortions and disadvantages. For example, autofluorescence of compounds, fluorescence quenching. You need reagents which are costly. Some reporters may interfere in other ways. And in contrast to that, MALDI-mass spectrometry per se is label-free and can nicely complement or even replace label-containing assay technologies. And therefore, we can see a hope now that we may eventually be able to screen some unscreenable drug targets for which current technologies have not been sufficient. To get into mass spectrometry profiling here, some of you may know the Bruker MALDI biotyper technology, in which complex mass spectra of bacteria are recorded, and based on the patterns representing parts of the proteome of those bacteria, together with multivariate statistical analysis, such as principal component analysis, one can distinguish very clearly and very easily and very quickly distinct bacterial strains and choose a proper treatment. For example, after sepsis diagnosis, one can choose the proper antibiotic. And a number of years ago, we were wondering why nobody is doing that with mammalian cells. After all, they also contain proteins and other things that can be analyzed by MALDI-MS. And so, years ago, a then PhD student, Bogdan, who is now with Sanofi, did a very simple experiment. He differentiated leukemia cells in cell culture, mammalian leukemia cells, and observed that the protein fingerprints, as you can see on that slide, are changing over time, and indeed they changed in a reproducible fashion. So it became clear to us that these scenarios, these fingerprints, can be interpreted in a meaningful way that would eventually lead to their utility in cell assays. And to give you a first example, we early case study at the action of HDAC inhibitors in cells, histone deacetylase inhibitors. And what these compounds are doing is to play a role in the regulation of chromatin compaction and thereby regulating RNA polymerase activity. So the HDAC enzymes lead to deacetylation of nucleosomes, histones, which is required for an open and transcriptable proteome. And therefore, the HDAC inhibitors play a role in this process by making sure that chromatin does not get converted into heterochromatin and instead stays open and it can be read. And as a consequence of HDAC inhibitor action, we can observe a polyacetylation of histones. And that was a starting point for Bogdan to dive into the first set of cell-based assays using MALDI-mass spectrometry. Contrast the very fast way of analyzing histone acetylation by MALDI-MS. Conventional methodologies that we and others have been using for many years always include various processing steps with reagents and additional processing steps such as cell extraction. And we had the idea about 6 years ago, that MALDI-MS could render all this processing obsolete and lead to a very fast procedure, also in sample preparation. And so bringing together, easy label-free analysis, we can show a first example on this slide. What you can see is a gel view of multi-mass spectra, which means mass spectra are basically flopped by 90 degrees, and you are looking at mass spectra from the top. And as the micromolar concentration of the drug SAHA are increased, you are moving down in that gel view. And so we are looking at multiple replicates at multiple concentrations and see a very clear concentration dependence of a mass shift observed in a regular interval at histone H4. And this mass shift, which is the consequence of polyacetylation, can easily and directly be converted into concentration response curves that can be used to derive IC50s from. So while that was still a manual assay at very low throughput, it clearly indicated that multi-mass spectrometry with a fast mass spectrometer can be useful for cell-based assays. Our next example shows some of the capabilities of current rapifleX MPP technology, and the example we chose were BCR-able protein kinase inhibitors. Now, many of you will know BCR-able as the driver oncogene in chronic myelogenous leukemia, CML, that is nowadays treated with the kinase inhibitor imatinib, as well as several second and third generation drugs. And whereas the immediate actions in terms of phosphorylations of substrates and so forth on the protein level have been well studied and have been known for some time, we wanted to find small molecule pharmacodynamic markers of BCR-able inhibitor action and set up the first higher throughput assay in cells. The general setup is as follows. Cell cultures are treated with drugs, either known drugs or test compounds, and all these processes can be automated. In our lab, we are using the rapifleX from Analytic Jena. And after drug treatment, the entire cell layer, or in some cases suspension cells, are carried over onto a MALDI steel target plate and mixed with a suitable MALDI matrix. And then very fast data acquisition happens using the rapifleX mass spectrometer, leading to a whole bunch of lipid or metabolite fingerprints, just like you have seen before on the protein level. And these fingerprints can be interpreted using software that we have written in-house, but also in many ways using the MPP software. And such processing goes in a phenotypic assay where we don't know what we are looking for. So we are in discovery mode. We want to find new markers of BCR-able inhibitor action. And these experiments were all carried out by David Weigt in our lab, who is now with Boehringer. You can see, again, similar to the MALDI biotyper, in principal component space, that as drug concentration increased, the overall pattern of these fingerprints changed. And that information can be analyzed to extract some masses, potential pharmacodynamic biomarkers, that show a concentration response, either a decreasing concentration dependence or an increasing, leading to a maximum and eventually downfall because of ensuing toxicity. And based on these masses that at this stage we don't know what they are, now we can derive IC50 values for various types of known and unknown BCR-ABL inhibitors. And obviously, most of us will be interested in identifying what these masses are or what the biomolecules underlying these masses might be. And for that purpose, we use a second Bruker machine, Bruker Solarix magnetic resonance mass spectrometer, which, based on very high molecular specificity and high resolving power, enables us to do structural elucidation studies and find out, for example, in this case, that the first potential biomarker at mass to charge 616.2 is really TB. So in these leukemia cells, we find a concentration-dependent increase in TB, which goes in line with redifferentiation of those leukemia cells. So that was not all that surprising. A second is a marker, which decreases with increasing drug concentration, was determined to be a particular variant of phosphatidylcholine, but interestingly, only a variant that was a potassium adduct. We did not find a strong trend like that for the sodium adduct, for example, and still can only speculate as to why that is. But very clearly, we can interrogate cell lysates and drug actions from a pharmaceutical perspective of finding new pharmacodynamic biomarkers or using these assays with newly discovered biomarkers to search for new compounds eventually. The second example that I want to present today is fatty acid synthase, which has played a major role in various indications in pharma industry, in particular also in oncology. Because it has been known for quite some time that many human carcinomas in which FAS-N, as we call it, fatty acid synthase, is overexpressed, are linked with poor prognosis in patients. And so it seems that blocking of the enzyme would be cytotoxic to cancerous cells because overall fatty acid biosynthesis would be blocked. So we test case to set up the first cell-based MALDI-MS spectrometry triggered mechanistic assay in which we are not broadly looking for new biomarkers as in the first case, but in which we are specifically looking for one marker that can be used as a mechanistic readout for a very specific enzyme inhibition. We picked malonyl-CoA, a very well-known metabolite, which is used in mammalian cells only by two enzymes, one that makes it and one that uses it, and the latter one being FAS-N. So if that biochemical reaction is inhibited by a suitable inhibitor, you would expect an accumulation in cells of malonyl-CoA. And that's what David focused in his, in which cell profiling, drug profiling was done in 96 cell cultures. They were transferred using automation systems to MALDI steel targets. MALDI matrix was applied. Data was acquired by the rapifleX MPP system, and then a single molecular marker, malonyl-CoA, was followed. And also in that case, various known commercially available compounds behaved as fatty acid synthase inhibitors. Some other ones, like orlistat, triclosan, which have been described in the literature, not so much. And since data we observed and obtained with commercially available compounds were so promising, and it really seemed as if we could get a direct readout with very little sample preparation and no reagents whatsoever, we teamed up with GlaxoSmithKline, the originator of one of our two compounds, and set up a great collaboration, really, in which they gave us some other of their patented compounds which had not been profiled extensively with new technologies and which had not been published yet. And we performed a double-blind study here, and when we reported back our results to GSK, they were quite thrilled that the IC50s determined in a very fast MALDI-MS-based assay matched their in-house data reasonably well, in fact, very well. And a very nice side effect here was that compound 1 turned out to be the most potent FAS-N inhibitor that had ever been published. A nice and important feature of MALDI mass spectrometry-based assays is that we are always recording full scan spectra. So we can still, even in situations like this one where we focus on malonyl-CoA, we can still interrogate the entire spectrum. And when doing so, David recognized that another metabolite which he, using magnetic resonance mass spec, found out was CDP choline, was changing dramatically in response to some compounds. And to just make a case, in the magnetic resonance mass spectrometry, the resolving power is so high that we can also use it as a control for all our other experiments. Why did CDP choline accumulate as well, and not only malonyl-CoA? Because CDP choline comes in biosynthesis through the Kennedy pathway from a very different angle. But looking at that schematic view here, it's clear that if fatty acid synthase is effectively blocked, or any of the other steps depicted here, we will not be able to make DAG, diacylglycerol, anymore. And so CDP choline has no taker anymore in the production of PC phosphatidylcholine. And so it's clear here that any inhibition of any of the enzymes going from top to bottom will lead to CDP choline accumulation, whereas only very specific inhibition of fatty acid synthase will lead to malonyl-CoA accumulation. And so it came as no surprise that also some of those compounds which had been described as fatty acid synthase inhibitors, like triclosan, they did inhibit CDP choline use, but they did not inhibit fatty acid synthase. And so clearly they must have different mechanisms. So in summary part, before I hand over to Melissa, cell-based MALDI-MS assays with the rapifleX MPP setup enable various new things. First, as we've seen in the BCR-ABL example, we can discover drug response markers in cell-based assays that may be able to turn into additional pharmaceutical assays. Also, it's a standardized label-free monitoring of enzyme inhibition directly in whole cells, needing very little sample preparation and then measuring by MALDI-MS. Automation is possible in our hands towards medium throughput screening capability, but of course something like that could also be taken to high throughput screening. And finally, because we are always looking at full scan spectra, now despite our intent to only look at malonyl CoA, we get a lot of different and additional information that we can interpret in a mechanistic sense. And with that summary, I would like to hand over to Melissa.
So my part of the talk now is about the automated label-free profiling of cellular drug uptake and drug-drug interactions. And I would like to get you all on the same page first and do a quick introduction of that topic. And first of all, this is the Pfizer's 4 pillars of efficacy. And this is a house consisting of 4 different pillars, which are equally important to ensure the efficacy of a drug. The first pillar is the exposure at the site of action. So we have to pose ourselves the question, does the drug really get where it should be in the end? The second one is the target engagement. Does it bind and really act on the target? Then we have got the third one, the functional pharmacology. Does that elicit a response? And the last one is the relevant phenotype. Does the drug perturb a phenotype? So you can imagine if one pillar out of this house would break down, then the whole house would break down. So every pillar is really important, and especially the first one, exposure at the site of action. So we have to figure out if the drug really gets where it should be. So now some simple ways about the lipid bilayer and how there is transport going on here in the cell. This is really a simple biologics, but I thought just to get you all on the same level here, some of you might not know about that yet. So just a short summary about that. We have got the passive transport, and this includes also the diffusion and the ease diffusion, which includes channels and carriers, like ion channels and glucose carriers. And then we have got the active transport, which is in a way dependent on ATP. And there we have got several things, like the first one is primary active transport. This is directly dependent on ATP. We, for example, have the sodium-potassium ATPase, which leads to an accumulation of sodium outside of the cell, and this needs direct ATP. Then we have got the secondary active transport, which can be carriers, for example, and also the transport proteins I will be talking about are in this group of the secondary active transporters. And here we have got a sodium-potassium ATPase, which helps to accumulate the sodium outside of the cell, and this accumulation of sodium then leads to an electrochemical gradient of sodium, and this leads to the transporter transporting the glucose inside of the cell. So for the membrane transporters, we have got several transport classes, transport protein classes. So we have got the SOC transporters, those are the solute carrier transporters. And then we have got the ABC transporters, which are primary active transporters most of the time. The SOC transporters are uptake transporters, and they include mitochondrial transporters, coupled transporters, exchangers, passive transporters, and vesicular transporters. And as you can see in this diagram here, we have got several sorts of transport proteins in a cell, and also water channels, ion channels, pump, and those ADC transporters. So what is now the reason to investigate those transporters? One severe thing that can happen is drug-drug interactions, which is an increasing burden for elder generation because we have got people tending to get older and older and tending to take several medications at the same time. And this can then lead to drug-drug interactions, meaning that one thing that can happen is a drug inhibiting a transport process, and then the other drug can't be transported in anymore. Or the next thing that could happen is this transport process being blocked, and then the drug cannot be taken up into an eliminating organ, for example, and then this drug accumulates in the blood, which can be toxic. So it is very important to investigate those drug-drug interactions, and they are dependent on those transport proteins, and therefore then an investigation of transport proteins is very important. So in detail, we are looking at the organic anion transporting polypeptide 2B1, short OATP2B1, which belongs to this class of SOC, solute carrier transport proteins, secondary active transporters. And it is an emerging transporter which is expressed throughout the body. So you can see that in this graphic here, it is expressed in the blood-brain barrier, ciliary epithelium, hepatocyte, small intestine, placenta, and also in the skeletal muscle. So we have really got it throughout the body, and therefore it is important to investigate it. Like Carsten just already mentioned, the current technologies for the transporter investigations are either fluorescence-based or radio-labeled, most of them. And we have got some positive and also some negative aspects for those techniques. The positive aspect about the fluorescence-based assays are that they are sensitive and that they are automatable. The positive thing about the radio-labeled experiments are that they are sensitive also and that they are simple to design. But of course they also have negative aspects, and a big one which affects them both is that they are labeled techniques. The fluorescence-based assay is also prone to false negatives and positives due to photonic interferences like autofluorescence and quenching effects. And the radio-labeled version, of course, there is the thing with the exposure to working personnel and the waste management, which is not that easy. So we have a possible counter screen for that, which is label-free and can also be done cell-based, and this is MALDI-MS. I think you all know now how MALDI-MS works, so I will not go into detail about that here, but I will go a bit into detail about the assay development. First of all, we asked ourselves which substrate are we using for this investigation we want to do. So we wanted to design an uptake assay through OATP2B1, the transport protein, and therefore we searched for a substrate. We built up already a fluorescence-based assay, and this was done with the fluorescent substrate dibromofluorescin, short DBF. But of course, as this is not a labeled technique anymore, we don't need the fluorescent substrate and can also use the non-fluorescent substrate estron-3 sulfate E3S. So we had a dynamic range of both compounds in the cell background and tested that with different matrices to decide on which substrate are we going to use for this assay. Then the next question is, how are we using the matrix and solvent composition? There are several matrices that you can use, and also the solvent depends on that. And therefore we used the best matrix from step 1 and optimized it in its composition. And last but not least, the analyte matrix ratio. And here, in a case of cell-based assay, this means that the cell number has to be optimized. The matrix composition, there are some important considerations that have to be kept in mind here. So for every case, the best matrix composition and application has to be found. There are about 4 criteria affecting this decision. First of all, we would like to have preferably a high signal of our substrate. Then, second of all, it should be usable with a pipetting robot system if you want to use a robot system. In our case, this was the cellular of Analytic Jena, and this one is not usable with acetone, so we have to adjust our plans to that. Then the third one is that there should be no background signal of matrix next to the substrate peak. And fourth, that there is a development of a uniform matrix layer. We have got different application ways for the different matrices, and I illustrated them on the right here. So we have got dry droplet application, sprayer, primer drop, or sandwich. And just to guide you a bit through that here, the dry droplet application means that we have got an analyte and a matrix, and we mix them together and apply them manually or with a pipetting system onto the target plate. Then we have got a sprayed application, which means that we apply the analyte on the target plate first, and then the matrix gets sprayed through a spraying device. Then we have also got a primer drop option, where we pipette the analyte onto the MALDI target plate and then let that spot dry, and also pipette the matrix on top of that dried spot. And last but not least, the sandwich application, like you would imagine a sandwich to be, first matrix, then analyte, and then matrix again. And this happens through coating the target plate with matrix first, and then applying matrix and analyte dry droplet on top. So our first experiment in this assay development was the decision for a substrate. And as I told you, we investigated both DBF and E3S as possible substrates for OATP2B1 and tested it in its dynamic range with different matrices. And you can see in panel A that E3S leads to a higher intensity that is detected compared to DBF, and also at lower concentration ranges of the substrate we can still see a quite good intensity of E3S. Therefore, we concluded to use E3S, and also in panel B you can see now which matrices did work for the detection of E3S, and we have got here DHAP matrix, which worked quite good, and also phenylcyanaminic acid amide. We also tried BHB and SDHB, but as you can see in the bad signal-to-noise ratio, those two matrices do not seem to work for this use case here. And when we are looking at the crystallization of the matrix in panel C, then we can also see that the crystallization of DHAP matrix and phenylcyanaminic acid amide are more homogeneous than for DHB and SDHB. What you saw before was a case of cell pellets. There was a substrate spiked into, and now this is a real cell treatment. So we treated the cells and then pelleted them afterwards, and are now analyzing really what is going on inside of the cell. And this led to the exclusion of the DHAP matrix, because when you look at panel B, you can see that there is a disturbing peak right next to the analyte peak, and we do not want that. This is why we excluded DHAP. And also when you're looking at the matrix crystallization, you can see that DHAP in the dry droplet format leads to a really strange crystallization, where we think that an automated measurement is not really possible, and because in the end we wanted to go for a dry droplet application because it is more possible to align with high throughput, we excluded DHAP. What you can also see in panel A is now the real proof of uptake into the cells already. So we used here phenylcyanaminic acidamide as the matrix, and in the red spectrum you can see the OATP2B1 overexpressing cells which were treated with the substrate E3S. Then we have got in the green spectrum the inhibited uptake. We use allotinib, short allot, as inhibitor because it is the standard inhibitor for this case, and this leads to a lot lower signal. And when we also compare that with the vector control, which is just an empty vector, we see a lot lower E3S intensity here. So this is our proof that we are measuring really the E3S that has been taken up through the OATP2B1 transport protein. What we then investigated was the influence of the application of phenylcyanaminic acidamide on the assay window. What we can see in panel A is that we tried those 4 different application types that I already explained to you, and saw that with primer drop, the first one PD, there was a high variation, and therefore we excluded it. Then we still have left dry droplet, sprayer and sandwich application, and as you can see in panel D, the sandwich application leads to a really low assay window of just a factor of 1.5 between the positive and the negative control E3S and E3S plus inhibitor treated. So we wanted to go either for dry droplet or the sprayer application because here the assay window is above 4. What you can also see is that we tried a normalization, and we did this on the deuterated standard of E3S, D4 E3S, which you can see in panel E. And when we apply the normalization, this leads to a reduction of the CV%. And therefore we concluded to go for dry droplet application because this is more of high throughput really. What is now important for the automation is that we are looking at the workflow here now. So we have got 2 separate workflows. The one we used for the development and optimization of the method, which you can see on the left. Here we used 24 well plates, and we did the incubation with the substrate directly in those 24 wells. And then we had to transfer them all manually into centrifuge tubes, centrifuge them, freeze them, resuspend them, spot them all manually onto a target plate, and then measure the data and evaluate it. What is better now with the automated version is that we seeded out the cells in 96 well plates, incubated them with the substrate, and could directly freeze them without centrifuging them anymore. Then the application of the cells and matrix is done with our pipetting robot, and then we measure the plates with a rapifleX using the MPP software, and then are ready to evaluate the data. This now looks not that clear, but to get that into numbers, for the left workflow, I think there is at least half a day needed for doing that, and the automated version, just the application is 2 minutes still. So this is a real time reduction here. So this workflow of the developed method can be seen here now. So we treated the cells with the pharmaceuticals directly in the 96 well plates and using the pipetting robot. Then we did the sample resuspension and the dry droplet matrix application also using the robot, the data acquisition with the rapifleX using MPP software. And this workflow can be then used for either characterizing the transport, so the evaluation of transport kinetics, or using it as a screening tool in the search for inhibitors. So this slide is about both transporter characterization and reproducibility check. And we investigated if the transport process is time and concentration dependent. What we can see in panel A is that it is concentration dependent, leading to a saturation of the curve after about 50 micromolar. And it is also a process that is time dependent, which can be seen in panel B. And we see a flattening of that curve here at about 5 minutes. This was also important to investigate because we had to define our optimal assay conditions, and they lie in the linear range of the uptake of OATP2B1. So therefore, we chose 10 micromolar E3S concentration and 2 minutes of incubation time for the assay. What you can also see here is the check of reproducibility, and we therefore investigated the standard inhibitor ilotinib and tried to do IC50 curves for it, and it came out as a really reproducible, and we were able to identify the IC50 values here, which did not differ a lot. So what is also very important to check is the reliability of the method. So we investigated the relationship of the intensity and the concentration of E3S. We spiked E3S to cell pellets in a concentration range and then investigated where the detection is still linear, and as we use 10 micromolar E3S in the assay, we wanted to see if this is still in the linear range, and as you can see in this picture here now, it is still linear, and therefore we could go on with that type of assay here. We were now able to really conduct our MALDI-MS assay, and we tested 300 top marketed drugs, and also some of the drugs were known to interact with the OATP2B1 transporter in their ability to inhibit this uptake of E3S through the transport protein OATP2B1. And we were able to identify out of those 367 inhibitors, meaning they had an inhibition of more than 50%. And as you can see in this table here, we had also some potent inhibitors with a PIC50 above 6, 14, and they are marked in red here. What you can also see is that we conducted 6 biological replicates, and it led to a mean standard deviation of 0.3 and a mean CV% value of 5.2, so a very reproducible assay. As we have already set up a fluorescence-based assay, we wanted to compare our results from the MALDI assay with the fluorescence-based assay. And first of all, we compared the MALDI-MS assay we did an intra-assay comparison, and this can be seen in panel A, and it led to a Spearman correlation coefficient of 0.86, so an acceptable reproducibility. And when we look at our fluorescence-based assay only, this has an intra-assay reproducibility of a Spearman correlation coefficient of 0.71. So now we compared the results of both techniques, which can be seen in panel C, and we had 47 overlapping hits. The MALDI-MS assay hit led to 67 hits in total, and 20 hits only were found with the MALDI technique. The fluorescence-based assay has 57 hits in total, and 10 hits were only found with fluorescence-based assay. And we then also compared the PIC50 value determination, can be seen in panel D, and here we have got a Spearman correlation coefficient of 0.44. So the question now is, why is there such a difference between those two techniques? And the most promising answer for that is that we used a different substrate for both of those methods. So as I told you, we used the dibromofluorescent, the fluorescent substrate for OATP2B1 in the fluorescence-based assay, and we used E3S in the MALDI assay, and there are a lot of papers indicating that with the use of another substrate, there are different inhibitors identified. What we then also wanted to do was compare our data with data that was really relying on E3S as a substrate. There was a study published by Carsten et al. Who used a radioactive assay to screen for inhibitors of E3S as a substrate. And we compared those two data sets and found a consistency of 83%. 12% of the inhibitors were only identified with our MALDI technique, and 5% were only identified by their radioactive assay. So this is a quite good correlation. And to conclude on that part of the talk, we developed and optimized the first cell-based unlabeled MALDI-MS uptake assay. We automated that assay and applied it to the evaluation of transport kinetics. We screened 300 marketed drugs and revealed 67 inhibitors in a very reproducible assay, so the CV% was under 6, and we compared it with the literature, and that revealed a good correlation of 83%. Carsten and I would both like to thank our great CeMOS team, and we would like to ask you if you want to do something together with us. Thanks for listening.
Thank you for that great presentation, Dr. Hopf and Melissa. Yes, we are now ready for some questions. Please use the Q&A box. I know that we already had some questions coming in early on, so I will go ahead and start with our first question. How do you separate the unbound drug to the drug bound to the target? For example, the specific receptor before introducing the sample to the MALDI.
So this is a question for me. We do not really separate this, but we do it like this, that we first incubate the cells with the drugs, and then we aspirate the medium, and we wash the cells with PBS. And after that, we freeze them, and then we resuspend them and only measure the cells in the end. So we do not really separate that, but the thing why we can say we are really looking at the transporting protein is that we have really defined by comparing the OATP2B1 overexpressing cell line with the empty vector control, and we have seen that the E3S uptake just happens through the OATP2B1 transport protein, and therefore we can really say that we are investigating an uptake, and therefore do not have to separate this unbound drugs.
Okay, thank you. Another question: which matrix do you use to detect masses lower than 500 Da?
Yeah, so maybe I take that.
Yeah, okay.
Maybe I take that one. You know, there are several different matrices, so you have to try a couple of them. Melissa has nicely outlined some of them that we've tried. You will find more information in that paper. There are more matrices out there. Most of them are actually for masses lower than 500. And you know, please, as Melissa pointed out, be aware of some interfering peaks. So you should always check a couple of matrices.
Okay, great. Thank you. Another question. Do you also perform the sample disposition and matrix mixing on the plate automatically, or do you still perform it manually? In case of automatically, do you do it with your classical liquid handler?
So for my part of the project, I can say that for the method optimization, we did that all manually, and that took a lot of time, and therefore we did the automation in order to really be able to perform that screen, and we used the cellular pipetting robot as our liquid handler for that, and now are applying the matrix and cells in a dry droplet way, via the liquid handling.
Okay, thank you. Another question. What solvent and composition did you use with the matrix?
So we used several, but in the end it turned out that 2.5 milligram per milliliter phenylcyanosinaminic acid amide in 70% acetonitrile worked best.
Okay, another question. How do you remove the cell culture media from the samples that you spot onto the MALDI target?
So this is, the cells are in culture. There we do the incubation with the drug. Then when the incubating time is over, we just aspirate them with a vacuum aspirator, and there is 8 canals on that, so we can just pretty quickly aspirate the cell culture medium. Then we wash that with PBS, and then we are ready to freeze the cells.
Okay, a couple people have just said great talk, so kudos to both of you. Great talk from one of the presenters as well, or from one of the people online. Their question is, since the inhibition can be substrate dependent, would it be possible to multiplex to use substrate in the same reaction? And I'm not sure what the symbols there are, so maybe you can see that on the screen there.
So theoretically, I would say yes, but maybe this might be a bit hard because several substrates for one transporter, I don't know if they then would compete in the end for the binding on this transport protein, but theoretically, when there is no problem with the binding proteins, binding to the protein, it would be possible because, of course, we are having a full scan spectrum, so we could look at different masses at the same time, but in the end, I don't know if this is biologically really possible.
Okay, it looks like a question's come in for you. Did you check for false positive fluorescent actives and false positives having similar weight in the case of MALDI? And if so, what was the outcome?
So we tested fluorescent photonic interferences, and we measured autofluorescence and quenching effects, and we really had some of those compounds showing an autofluorescence, so no autofluorescence but quenching, and I think there were 3 out of those 300 that really showed a quenching effect, and therefore we took them out in the comparison with the MALDI assay, and the false positives in case of MALDI were not checked.
Okay, another question here. Are the cells lysed before mixing with the matrix, and how do you lift adherent cells from the assay plate to mix with matrix?
So there is no lysation step in our protocol, and you do not really have to lift those or trypsinate them in case of the manual handling. Of course, you do have to trypsinate them and then transfer them to a centrifuge tube and centrifuge them to get a pellet. But in case of the automated assay, you just have your adherent cells sticking to your 96 well plate, and then when you put the water on there as a resuspension solvent, then the cells will break up, and you mix it together with the matrix, and what you're analyzing then is what was inside of the cell. So it is really a resuspension step, And I guess there will also, in the end, land some of the cells on the target plate, but what is the most important thing is that we are really analyzing what was inside of the cell, because we did a breakup by using the water as a resuspension solvent.
Okay,
yeah, so if I may just
yes, please.
specify to that, you know, so there is no dedicated lysis step where you say, now we're doing lysis. You know, so it all happens in passing. You know, hypotonic lysis, you have 70% acetonitrile in there. So there will be some lysis, but it's not a dedicated lysis step, which is part of the beauty of it.
Thank you. Thank you for the added commentary. Also, question, can protein aggregates be analyzed using this screening method?
Yeah, that's an interesting one. Maybe I take that one. The answer is no, but that would be something we'd be really keen on checking out. No, we haven't done it. I'm not aware of anybody having done it.
Okay. Another one with a great talk to you both. And what sort of extraction did you carry out on the cells?
Yeah, so I think we also got that in the last question, some of it, because we really did not a real extraction, but we just resuspended the cells with the water and then applied them onto the target plate.
Okay, thank you for that. Have you checked the limit detection concentration of the drug?
So we did not do that for the set of the 300 marketed drugs, but we also do not need to do that, because in the end we're only looking at the M% of the E3S. So we do not look at the drugs itself, but on the what outcome they have on the uptake of the substrate. So we are just looking at the E3S, and therefore we do not have to check the limit of detection of those drugs.
Okay, another question. Is it possible to detect picomolar concentrations of drugs with this equipment?
Yeah, maybe that's one for me. So most likely no. But I'm also not clear why you would have to do that. You know, because if you're talking about compound uptake assays and you want to really measure the compound which has been taken up, you would have to optimize your MALDI method for every new compound. So that would not be a most sensible method. What Melissa has done instead is, you know, an indirect measurement to optimize a method for a substrate and then look for inhibition. You know, setting up a compound uptake assay itself might be difficult because, you know, 300 compounds all are different, would require different MALDI matrices. So it's not just about concentration of compound. It would probably not be useful.
Okay, another question. In what ways is MALDI-MS superior or complementary to radio-label fluorescence-based assays?
So I think we pointed that out already, that the big advantage about MALDI is that, of course, it is unlabeled, and therefore we don't need a substrate which is either fluorescent or radio-labeled, and so we can use any substrate we are thinking of. And this is really the big advantage about that. Of course, it is also a fast technique and is automatable. And the fluorescence and the radio-labeled techniques do have some disadvantages, like the photonic interferences in the case of the fluorescence-based assays. And of course, for the radio-labeled versions, the exposure to the working personnel. So there are a lot of advantages for MALDI.
Okay, hopefully this wasn't already, sorry if some of these have already been asked. I'm having a hard time keeping up with them, but how quantitative are the results?
Well, maybe I take that. How quantitative are they? as you've seen in most cases, quantities that we're most interested in are IC50s. And our main focus has always been reproducibility of IC50s after curve fitting. And that turned out to be very reproducible, so we were very happy with that. Now, quantitative results as in IC50s. If you're talking about quantitative as in signal intensity, then it certainly helped a lot to include internal standards. So for any practical purposes, I would say it is necessary to include, for example, stable isotope labeled internal standards to cope with the variability inherent to the MALDI process. But with that done, surprisingly, results turned out to be quite quantitative, and it is well accepted now for biochemical assays. Many major pharmaceutical companies are running them even for high throughput screening, that MALDI assays per se can be quantitative for industrial purposes. And I believe the same can be said with a little more optimization also for cell-based MALDI assays.
Okay, this is going to come back to Melissa. They wanted a little more clarification. They're not sure they followed what Melissa meant by vacuum aspirating the cells to remove the culture media. Could you elaborate on the process?
So we have got that 96 well plate, and the cells are in the cell culture medium inside of this 96 well plate, and then we do the drug incubation there, and then we have got a vacuum aspirator, and it has 8 channels, and therefore can use one row of those 96 well plates at once, and then just suck that cell culture medium. So remove it by using this vacuum aspiration. And then when that is all gone, the cell culture medium, it is once again washed by using a multi-channel pipette, using PBS, and then once again removed by vacuum aspiration, and then the cells can be frozen.
Okay, another couple of questions here before we run out of time. Is there a perspective for automation, and is further miniaturization of the assay conceivable?
Definitely yes. We do not have a device yet that would help the automation in total. So we don't have a device helping us to really put the plates in. So we are doing that all manually now. So this would be another part of the automation process. And of course, there is the possibility to use 1,536 well plates, so the multi-target plates instead of our 384. So this is just for the miniaturization of that possible.
Okay,
and we're currently actively working on miniaturization in particular.
Okay, thank you. Another question here. Maybe we have time for one more. How does MALDI-MS aid the discovery of candidate drug responses? I'm not sure if that was already asked.
Now, maybe to just conclude our session, as I've shown in those examples, it is particularly powerful to look at potential response markers that people typically are not looking at. For example, lipids, metabolites, where you would need mass spec assays in any case because it's almost impossible in any other way. So we've shown some nice examples, and now are working on some other cases with some partners right now. So I firmly believe that it is at least a complementary method to other approaches using LC-MS.
Thank you. A lot of great questions, and we've pretty much run out of time. If we didn't get to your question, we'll certainly answer that offline. I definitely want to thank the presenters for such a great presentation and you, the audience, for your participation in our webinar today. If you are interested in viewing future webinars, and obviously with the COVID crisis here, we're doing a lot of webinars, so we do have quite a lineup also still to the end of the year. You could visit our website at www.bruker-webinars.com. Please, everyone, stay safe. And again, thank you all for attending today's webinar.
Thank you.